This is our Chapter 11 outline, where we're looking at inference for distributions of categorical data, as opposed to quantitative data, which is what we looked at in Chapter 3. So a guiding question for the whole chapter is we're looking at how can we make inferences by finding patterns and correlations in categorical data. Remember Chapter 3 was quantitative, and we looked at R and R squared to find the goodness of fit. So also remember that we had two-way tables that measured two variables way back in Chapter 1, and we used them to create conditional distributions and marginal distributions. So some of this is going to be similar to what we've gone over in Chapters 1 and 3. Some of it's going to relate to probability. So a quick reminder that independent means the outcome of one event does not affect another. So if two events are independent, call them A and B, the probability of A is the same even if B has already happened. Remember, this indicates here that the probability of A occurring given that B already happened. So that means this event, B, has happened. The probability of A is still the same even if B has happened or if it hasn't. And vice versa, the probability of B is the same whether or not A has occurred. That's the definition of independence mathematically. So our first lesson, we're going to look at 11.1, the chi-square goodness of fit test, and how to calculate the chi-square statistic. So for biology students, you've done some of this before, although we may get more into the concept of chi-square, the actual distribution, what it looks like, the graph, and some other aspects of it, like checking conditions that you may not have done. So for 11.1, we're looking at what is a chi-square goodness of fit test? How is it different from determining the amount of correlation as we did in Chapter 3? And also keep in mind, how does it help us make inferences by finding patterns and correlations in categorical data? So in 9 and 10, we looked at significance tests about population parameters, such as proportions or differences between population means. Now we're looking about tests about distributions of categorical variables or relationships between two categorical or more categorical variables. So prior, an alternative hypothesis could be one-sided or two-sided depending on the question we were trying to answer. But now in this chapter we're going to be always looking at categorical data. So the alternative hypothesis will always be that the null hypothesis is not correct. So it would always be a two-sided test. Never be a one-sided test. So keep that in mind as we go forward. We did a lot of one-sided tests in the last two chapters. When stating your hypothesis, you're going to be looking at observed counts and expected counts, and your hypothesis is going to oftentimes use symbols. You may use the letter P for a proportion, for example, of candies from a machine that are a certain color, red, blue, yellow. Remember, don't use p-hat, as that indicates a sample statistic, and our null hypothesis is making a claim about the actual parameter, not the sample statistic. A one-way table is often used to display the distribution of categorical variables. We'll talk about that in class and see what you think. Chi-square goodness of fit test tests this null hypothesis that a categorical variable has a specific distribution, and that would be the one that's specified in the null hypothesis. So we're comparing what we would expect to what we observe based on the assumption that the null hypothesis is true. So expected counts for any category can be found by multiplying the proportion of the population distribution in that category by the sample size. So if I expect that 10% of a bag of 100 candies are red, then my expected count would be 0.1 times 100, or 10. I'd expect to have 10 of them. If I expected that 11% would contain it, then it'd be 0.11 times 100, or 11 of them. Now our chi-square statistic, we use this symbol here that looks somewhat like an X for chi-square. And what it is all the observed, meaning what the actual count we get, minus our expected value, and then we square that and divide it by the expected value. So the difference between the observed and expected squared divided by the expected. And then keep in mind this summation symbol means you would add them up. You'd do that for every category. So if we're talking about candies, as I did before, you would do that for the observed minus expected red over the expected red. You'd square the numerator there, observed minus expected squared. And then you'd go on and do the same thing for orange candies, the observed orange candies minus the expected orange squared over the expected orange, and add those numbers up for each category. That would be your chi-square statistic. This is the sum over all possible categories, would be the chi-square statistic. So the big difference here The degrees of freedom take on a different meaning here for a chi-squared statistic. They're actually dependent on the number of categories. So categories being red, yellow, green, blue for candies, like I just said. So if we had red, yellow, green, blue as our 4 kinds, that's 4 categories. Our degrees of freedom would be 4 minus 1 or 3. So it's not dependent on sample size as it was with the t-distribution. As before, though, large values of chi-squared are evidence against the null, just like a large, we discussed a large z-score or a large t-score test statistic was evidence against the null hypothesis. So if we had one well over 3, then we know that there's very little chance that the null hypothesis was true given our sample data. So large values would be good evidence against the null hypothesis, as our observed counts are further away from our expected counts if the value of chi-square is large. If the value of chi-square is small, then that means our observations are close to what's expected, and that means they're supporting the null hypothesis, which the null hypothesis would be the expected values for each category. So to do this, we'd find a p-value for the chi-square statistic we get. When we say the chi-square distribution, we're talking about it's an approximation to the sampling distribution of the statistic chi-squared. Now our 3 conditions are the same except for one. So we need an SRS just as before. If an experiment, we need random assignment. So we say RA for that sometimes, random assignment. Remember that helps account for lurking variables. That also helps account for them by creating 2 roughly equal groups in order to account for them. It lets us determine cause and effect. So if we have a treatment and we have a control group, it lets us determine that the treatment's effective. Whereas the random SRS lets us apply our results to the population from which it came. Now our second condition is a little different. It's large sample size. All expected cell counts are at least 5. That means the number of in our sample times the probability of each category. When you multiply that out, it would be at least 5. And then finally the independent. So this is the one that's different. So take a look closer over this. And then the independent condition is our final one, which is just the same as before. Sampling without replacement, check the 10% condition. Also, one outcome should not affect another. Like in the Brandon Belt question, one at bat should not affect another. So if we had an evenly distributed 4 colors of candy, 25% would be our expected proportion of each. If we had an equal number of blue, red, yellow, green candies, so 25%, I need to have 25% times my sample size be at least 5. That would mean I need to have at least 20 candies that I'd be sampling total from to calculate the observed minus expected candies, because 25% of 20 is 5. So that's what's meant by this condition. Now, if a test is statistically significant, we do a follow-up analysis that compares the observed and expected counts and looks for what's the largest component of the chi-square statistic, meaning which observed count is furthest from the expected count. Now this example is on page 680 in your book, and it goes through how to calculate it. So here we have the observed minus expected candy squared over the expected, and then we add it up for the next category, the next, and so on. We end up having 1, 2, 3, 4, 5, 6. That means there must be 6 categories: blue, orange, green, yellow, red, brown. So our degrees of freedom here would be 6 minus 1 or 5. Remember, it's the number of categories minus 1. So turn to this example in your book. We have some discussion on it here. So these are essentially the distance from the expected count. We square them so they all end up positive. All distances are positive. It's a similar idea to standard deviation, right? Except for now we're measuring the distance from what we'd expect as far as probability goes. So it's never going to be negative, it's always going to be positive. Large, because of that squared sign in there, and you're adding them all up. Large values would be strong evidence against the null. Small values are weaker evidence against the null and suggest that the data are consistent with our null hypothesis. And to determine the relative size, we're going to have to know how many categories there were to determine degrees of freedom, compare it to a chi-square curve, which looks different than our normal curve. and we're looking to see how it would vary in repeated random sampling to see if we could get a chi-square statistic like that by chance, or whether it's evidence against the null hypothesis. Okay, so take a look in your book, look over the summary for 11.1, read over the lesson, and here's your multiple choice question. How do we determine the degrees of freedom for a chi-square statistic? There's your 5 possible answer choices. Please pause now to answer the multiple choice and the free response.